Hello everyone, and welcome to our presentation. Today, we will be exploring the structure, function, and mechanics of the respiratory and circulatory systems. The title of our session is Exploring the Structure, Function, and Mechanics of the Respiratory and Circulatory Systems, with a focus on providing a comprehensive overview of cardiopulmonary anatomy and physiology. Over the next few slides, we'll delve into how these systems work together, their key components, and their vital roles in maintaining our health. I'm excited to have you here and look forward to an engaging and informative session. Let's take a closer look at the comprehensive overview of cardiopulmonary anatomy and physiology. Starting with the role of the respiratory system, its primary function is to bring atmospheric gases into contact with our blood to facilitate gas exchange. This process is vital because it maintains our oxygen supply and helps remove carbon dioxide from the body. When we talk about ventilation, we're referring to the movement of air into and out of the lungs. Respiration, on the other hand, involves the actual exchange of gases. External respiration happens between the air in our lungs and the blood, while internal respiration takes place at the tissue level, between the capillaries and our body's cells. Understanding these processes helps us see how efficiently our lungs work. At rest, they operate with minimal muscular effort, yet they have reserve capacity. This means they can adapt to increased oxygen needs during physical activity. The gas exchange mechanisms are crucial. External respiration ensures oxygen moves from the air into the blood, and internal respiration guarantees that oxygen reaches tissues while waste gases are removed. Together, these processes form the foundation of how our cardiopulmonary system sustains life and supports activity. Let's start by looking at the anatomy and function of the upper airways. This slide provides a comprehensive overview of the cardiopulmonary anatomy and physiology, which is essential for understanding respiratory health and disorders. First, consider obstructive sleep apnea. It occurs when the muscles in the pharynx relax too much during sleep, leading to airway obstruction. The main treatment for this condition is CPAP, or continuous positive airway pressure, which keeps the airway open throughout sleep. Next, we have epiglottitis. This is a serious condition characterized by symptoms like drooling, a muffled voice, and a classic thumb sign seen on X-ray. It's a medical emergency because it can quickly block the airway, requiring urgent management to secure breathing. Moving on to the larynx, often called the voice box. It contains the vocal cords and key landmarks such as the thyroid cartilage and the epiglottis. The larynx is crucial not only for producing voice, but also for protecting the airway during swallowing. The anatomy of the vocal cords and larynx is complex, but vital. Their proper function ensures that speech is clear and that the airway remains protected from food or liquids entering the lungs. Now, let's discuss the pharynx, which is divided into the nasopharynx, oropharynx, and laryngopharynx. These segments work together with muscular control to prevent aspiration. That is, they help keep food and liquids from entering the airway. Reflexes involving the glossopharyngeal nerve for sensation and the vagus nerve for motor control play a key role in maintaining airway protection. Finally, the nose, or nasal cavity, is the primary route for breathing at rest. Its main functions include warming, humidifying, and filtering inspired air. Key structures like the nares, nasal septum, and turbinates increase the surface area and improve these functions, making sure the air reaching our lungs is clean and at the right temperature. Overall, the upper airways conduct air efficiently from the environment to the lungs, but they do not participate in gas exchange themselves. Their role is crucial in ensuring that air reaches the lungs effectively and safely, which ties into the broader theme of respiratory health and disease management. Let's take a closer look at the lower airways and conducting airways. The first key point is the isothermic saturation boundary, or ISB. This boundary is located just below the carina, which is the point where the trachea divides into the main bronchi. At this boundary, inspired gases reach body temperature and are fully humidified, which is essential for optimal respiratory function. Maintaining this boundary helps protect the lower airways and ensures efficient gas exchange down the line. Moving on to the main bronchi, the left main bronchus is narrower, longer, and angled at about 45 to 55 degrees. This anatomical feature actually helps reduce the risk of aspiration on the left side. Because of its shape and length, foreign material is less likely to enter the left bronchus compared to the right. In contrast, the right main bronchus is wider, shorter, and more vertical, with an angle of roughly 20 to 30 degrees. This structure makes it more prone to aspiration, and it's also the typical pathway for right mainstem intubation during airway management, which is an important consideration during procedures. Now, the carina itself is a critical landmark. It marks the bifurcation point of the trachea at roughly the C6 to T5 vertebral levels. Recognizing the carina is vital because it guides us in airway placement and helps prevent complications during intubation. Finally, the tracheobronchial tree branches dichotomously. That is, it splits into two, at the carina, into right and left main bronchi. These bronchi then continue dividing into lobar, segmental, and subsegmental bronchi, creating a highly organized tree that distributes air efficiently throughout the lungs. Understanding these structures and boundaries is fundamental for effective airway management and respiratory physiology. Let's take a closer look at airway histology and mucociliary clearance. First, it's important to understand that aside from the epithelial cells, there are several non-epithelial cells like eosinophils, neutrophils, and mast cells. These cells play critical roles in various airway conditions. Eosinophils are often involved in allergic asthma, neutrophils respond to bacterial infections, and mast cells are key players in bronchoconstriction. Now, environmental factors such as smoking, pollution, and dehydration can significantly impair mucociliary clearance. When this clearance is compromised, it can lead to or exacerbate diseases like chronic bronchitis, cystic fibrosis, and asthma. So, maintaining a healthy environment is crucial for airway health. Moving on, the mucociliary clearance system is quite efficient. Around 250 cilia are present on each epithelial cell, and they beat approximately 1,300 times per minute. These tiny hair-like structures work together to move mucus, which is organized into two layers, a thick gel layer and a watery sole layer, facilitating effective clearance of debris and pathogens. The lamina propria underneath contains essential support components like smooth muscle, elastic fibers, blood vessels, and nerves. These elements help maintain the structural integrity of the airways and support their function. Lastly, the conducting airways are lined by a pseudostratified columnar epithelium, which forms the respiratory mucosa. This lining includes goblet cells that produce mucus and submucosal glands, secreting about 100 milliliters of mucus daily. This mucus traps particles and pathogens, working hand in hand with the cilia to keep our airways clear and healthy. Let's take a closer look at the alveoli and the gas exchange membrane. First, alveolar macrophages play a crucial role in lung health. They're responsible for clearing debris and microbes that reach the alveoli. Think of them as the lung's main defense system, especially important beyond the terminal bronchioles to prevent infections. Moving on to the cells within the alveoli, type II cells are fascinating. They are polygonal in shape and secrete surfactant from structures called lamellar bodies. This surfactant is vital because it reduces surface tension within the alveoli, helping to keep them stable and preventing collapse during exhalation. In contrast, type I alveolar cells are flat and cover most of the alveolar surface. Their primary role is to facilitate gas exchange, oxygen in, carbon dioxide out. Their thin, flat shape allows for efficient diffusion of gases across the alveolar capillary membrane. Speaking of that membrane, it's remarkably thin, less than half a micrometer thick. This thinness makes it highly permeable to gases, which is essential for quick and efficient diffusion. However, it's selectively permeable, meaning it doesn't allow liquids to pass through easily, maintaining the proper environment for gas exchange. Now, considering the scale, our lungs contain about 300 million alveoli, providing an enormous surface area. between 50 and 100 square meters. To give you a sense of size, that's roughly the area of a tennis court. The alveoli themselves are tiny, with diameters ranging from 100 to 300 micrometers, and each is surrounded by pulmonary capillaries just wide enough for red blood cells to pass through. All these features together create a vast, highly efficient surface for gas exchange, ensuring that oxygen reaches our blood and carbon dioxide is expelled effectively. This intricate design is fundamental to the overall function of our respiratory system. Let's take a closer look at the anatomy and physiology of the cardiopulmonary system. The lungs are conical organs, with an apex that extends above the clavicles and a concave base that rests on the diaphragm. The left lung has a unique feature called the cardiac notch, which makes space for the heart, and it also has a lingula, which corresponds to the right middle lobe. Focusing on the lung surfaces, the costal surface is smooth and lies next to the chest wall, while the mediastinal surface is concave and contains important structures like the heart, great vessels, esophagus, trachea, and bronchi. Now, moving on to the pleural membranes, the visceral pleura directly adheres to the lungs, and the parietal pleura lines the chest wall. Between these two layers is the pleural space, which contains lubricating fluid. This fluid creates a negative pressure, or negative intrapleural pressure, which is essential for allowing the lungs to expand smoothly during breathing. Understanding this, we see how critical the integrity of these membranes and the pleural space is. When fluid accumulates in this space, it leads to a condition called pleural effusion. On imaging, this can be seen as a blunting of the costophrenic angle. Another important condition is pneumothorax, which involves air entering the pleural space. This disrupts the negative pressure and causes the lung to collapse, impairing normal breathing. So, in summary, the anatomy of the lungs and chest wall, along with the pleural structures, plays a vital role in normal respiration and can be affected by various clinical conditions that we'll explore further. Now, let's discuss the blood supply and lymphatics of the lung. First, the pulmonary circulation plays a crucial role in gas exchange. Blood from the right ventricle is carried via the pulmonary arteries to the alveolar capillaries, where oxygen is absorbed and carbon dioxide is released. The oxygenated blood then returns to the left atrium through the pulmonary veins, completing this vital cycle. Alongside this, the bronchial circulation originates from the aorta. It supplies oxygen-rich blood directly to the airway walls and lung tissues beyond the alveoli, ensuring that all lung structures receive proper nourishment. This system works in tandem with the pulmonary circulation to keep the lungs healthy and functional. Moving on to the lymphatic system, it handles a significant amount of fluid, about 30 liters daily, from the capillaries. It filters this fluid and returns approximately 3 liters per day via lymphatics to prevent pulmonary edema, which is swelling caused by excess fluid. A key point to remember is the anatomical shunt that occurs in the lungs. Some oxygenated blood from the pulmonary veins mixes slightly with deoxygenated bronchial venous blood. This mixing slightly lowers the oxygen levels in the systemic arterial blood, which is an important consideration in understanding lung physiology. Overall, these systems, the pulmonary and bronchial circulations along with the lymphatics, work together to maintain efficient gas exchange, tissue nourishment, and fluid balance within the lungs. They are fundamental to respiratory health and function. Let's take a closer look at the nervous control of ventilation and the anatomy of the thorax. First, the movements of the rib cage, such as the bucket handle and pump handle motions, are crucial because they increase the thoracic volume. These movements create pressure gradients that are essential for airflow, allowing us to breathe efficiently. The thorax itself is made up of several key structures: the rib cage, intercostal muscles, vertebrae, sternum, and diaphragm. It's divided into the left and right pleural cavities, which house the lungs, and the mediastinum, the central compartment containing vital structures. Now, moving to the nerves involved, intercostal nerves innervate the intercostal muscles, which are vital for the mechanics of breathing. These muscles contract and relax to help expand and compress the thoracic cavity during respiration. The diaphragm, which is the main muscle of respiration, receives its motor innervation from the phrenic nerves, originating from C3 to C5. When there's injury above C3, it can lead to paralysis of the diaphragm and other ventilatory muscles, severely impacting breathing. Finally, regulation of ventilation involves both voluntary and involuntary control. The somatic nervous system allows us to consciously control the diaphragm and chest wall muscles, especially during activities like talking or deep breathing. Meanwhile, the autonomic nervous system manages involuntary control over smooth airway muscles, ensuring continuous, automatic regulation of our breathing without conscious effort. Understanding this intricate neural control and anatomy helps us appreciate how our respiratory system functions seamlessly to sustain life. Let's take a closer look at the functions of the diaphragm and intercostal muscles. The diaphragm, as you know, is the primary muscle responsible for breathing. During normal, quiet breathing, it moves about 1.5 centimeters, which generates roughly 500 milliliters of tidal volume, the amount of air we typically breathe in and out. When we exert ourselves, the diaphragm can move much more, up to 6 to 10 centimeters, significantly increasing the thoracic volume and allowing us to breathe more deeply. Now, in the context of diseases like COPD, the diaphragm can become flattened and even move paradoxically, which means it doesn't move as it should. This impairs ventilation efficiency and makes breathing more difficult. Understanding these impairments helps us grasp how respiratory diseases affect overall function. Moving on to accessory muscles, these become especially important during increased work of breathing. Muscles like the scalenes, sternomastoids, and pectoralis major kick in to assist with ventilation when more effort is needed. Additionally, the abdominal muscles play a vital role during forced expiration. They help push air out more effectively, especially during exertion or respiratory distress. Regarding the intercostal muscles, the external intercostals elevate the ribs during inspiration, mainly during times of increased demand. The internal intercostals have a dual role. Some fibers assist with inspiration, while others help with expiration. Their coordinated action helps expand and contract the chest cavity efficiently. Finally, let's touch on the structure of the diaphragm. It has two parts: the costal part, which originates from the ribs and sternum, and the crural part, originating from the lumbar vertebrae L1 to L3. When the diaphragm contracts, it pulls the central tendon downward, lowering the dome and lifting the ribs. This movement increases the thoracic volume during inspiration, facilitating airflow into the lungs. Understanding these muscles and their functions gives us a comprehensive view of how breathing is orchestrated and how diseases can disrupt this delicate process. Let's take a closer look at the mechanics of ventilation, focusing on resistance and elasticity. These are fundamental for understanding how our lungs inflate and how air flows through our respiratory system. Grasping these principles is key to evaluating respiratory health and function effectively. When we talk about lung volumes and capacities, we're referring to measures like tidal volume, which is the amount of air we breathe in and out during normal respiration, residual volume that remains in the lungs after exhaling, and inspiratory and expiratory reserves, which are the extra amounts of air we can inhale or exhale beyond normal breathing. Vital capacity and total lung capacity give us an overall picture of lung size and function, and these measurements are essential tools in respiratory assessment. Now, regarding the movements involved in breathing, the rib cage and diaphragm work together, but they can also act independently. During quiet, restful breathing, the diaphragm is the main driver, contracting to create the negative pressure needed for air to enter. However, during deeper or forceful breaths, the rib cage plays a larger role, expanding to accommodate greater lung volume. Let's also consider the pressure gradients that drive airflow. Air moves from areas of high pressure to low pressure, and this movement is facilitated by transrespiratory pressure differences. Transpulmonary pressure keeps the alveoli open, ensuring effective gas exchange, while transthoracic pressure reflects the recoil of the chest wall, aiding in expiration. Finally, when it comes to lung inflation, resistance plays a big role. There are two main types: frictional resistance, caused by airflow friction within the airways, which is dynamic and varies with airflow rate, and elastic resistance, which results from the elastic properties of lung tissue and the chest wall. Elastic resistance can be both static, present even at rest, and dynamic, changing during breathing cycles. Understanding these concepts helps us appreciate how complex and finely tuned our respiratory system truly is. Let's take a closer look at the pressure-volume curve of the lung. It's actually nonlinear, which means it doesn't follow a straight line, and it exhibits hysteresis. In simple terms, this means that more pressure is required to inflate the lung to a certain volume than is needed to deflate it back down to the same volume. This hysteresis is an important concept because it reflects the different energy requirements during inflation and deflation. Moving on to alveolar recruitment, this happens early during inspiration. As we start to breathe in, closed alveoli begin to open up as transpulmonary pressure increases. This process is crucial for effective lung function because opening more alveoli allows for better gas exchange. Think of it as unlocking more rooms in a house to let in air more efficiently. Now, lung compliance is a key measure here. It's calculated as the change in volume over the change in pressure, or delta/delta. Normal compliance at functional residual capacity is about 0.2 liters per centimeter of water. This tells us how easily the lungs can expand during breathing. When compliance is high, as in emphysema, the lungs tend to inflate easily but don't recoil well. That can lead to problems with exhaling properly. Conversely, low compliance, like in fibrosis, means the lungs are stiff and require higher pressures to achieve the same inflation, making breathing more effortful. Understanding these compliance differences is vital in diagnosing and managing various lung diseases. It gives us valuable insights into the mechanical properties of the lungs and how they function during respiration. Finally, by studying static pressure-volume relationships and compliance, we gain a deeper understanding of lung mechanics, which helps us tailor treatments and interventions to improve patient outcomes. Surfactant plays a crucial role in reducing fluid leakage from the capillaries, which helps keep the alveoli dry. This dryness is essential because it allows for efficient gas exchange, ensuring that oxygen and carbon dioxide can move freely between the lungs and the blood. When surfactant is deficient, as seen in premature infants, it can lead to respiratory distress syndrome. Without enough surfactant, the alveoli tend to collapse at low lung volumes, making it much harder to breathe and oxygenate the blood properly. Pulmonary surfactant is secreted by specialized cells called type II alveolar cells. Its main function is to lower surface tension within the alveoli, which helps stabilize alveoli of different sizes. This stabilization reduces the effort needed to breathe and prevents alveolar collapse, also known as atelectasis. Now, to understand how alveoli stay open, we can refer to Laplace's law, which states that the pressure needed to keep an alveolus open is proportional to the surface tension divided by the radius. This means smaller alveoli, with a smaller radius, require higher pressure to stay open if the surface tension remains the same. This explains the challenge smaller alveoli face in maintaining stability. Finally, surface tension from the liquid lining the alveoli naturally tends to cause them to collapse. This elastic recoil is a key part of how our lungs return to their resting shape after inhalation. Surfactant helps counteract this tendency by lowering surface tension, making it easier for the lungs to expand and contract efficiently during each breath. Let's take a closer look at flow patterns and airway resistance distribution. Diseases like asthma and chronic bronchitis can cause an increase in resistance within the small airways. When resistance goes up, it can significantly impact airflow and overall breathing efficiency. This is especially important because even small changes in resistance can make a big difference in how well we breathe. Now, considering the contribution of different parts of the airway, The upper airways are responsible for roughly 50% of the total airway resistance when at functional residual capacity. Although small airways, those less than 3 millimeters in diameter, contribute less overall, they become quite significant in conditions like asthma, where resistance can sharply increase. Breath sounds can tell us a lot about airflow patterns inside the airways. For example, tracheal sounds are typically harsh and high-pitched, reflecting turbulent airflow in the trachea, while vesicular sounds are softer and low-pitched, indicating more gentle airflow in the smaller peripheral airways. An important principle to remember here is Poiseuille's law. It tells us that resistance is inversely proportional to the fourth power of the airway radius. So, if the radius halves, the resistance doesn't just double, it increases 16-fold. This exponential relationship explains why small changes in airway size can have such a big impact on airflow. Finally, airflow within the airways can be laminar, turbulent, or transitional. Laminar flow is smooth and organized, typical in smaller, less constricted airways. On the other hand, turbulent flow is chaotic, noisy, and often occurs in larger or more constricted airways, especially during increased airflow demands. Recognizing these flow patterns helps us understand the mechanics of breathing and how diseases alter these processes.